afterwards to review, and in case people who missed it also they'll be able to just email us and be able to get the recording. Okay, so today, everybody, welcome to our presentation on quality results, how to obtain quality results with FCS Express. This presentation is going to be about 45 minutes to an hour, depending on how many questions we get. And this is not a presentation about how to use FCS Express or learning different features about FCS Express or step-by-step instructions on how to do things in FCS Express. If you're interested in that, we do have many webinars coming up as part of the current webinar series. You can go to our webpage. If you go to our homepage at denovosoftware.com and you scroll down a little bit, you will see upcoming live webinars. And you can see that we have many. Getting started with FCS Express Image is the next one. FCS Express Flow is coming up shortly afterwards. Webinar on spectral cytometry, how to do stain index, and many others are coming up. These are more going over specific features and how to do different things in the software. Today's presentation is more about how to use FCS Express to support your quality processes that are required in a validated lab, whether that's a GXP lab or a clinical laboratory. So we're going to show a lot of examples of how FCS Express can be used and is used by our customers to streamline and make their all of the validation burden much more straightforward, much easier to handle, and much more rigorous than the processes they're doing now. Once again, if you just joined us, if you do have any questions during the presentation, there is a question and answer tab in the GoToWebinar control panel. Feel free to open that up and type any questions there, and we'll get to them right away. Okay. So some terminology before we start. Assays in your clinical lab need to be validated. You need to demonstrate that that method is suitable for intended use, and you need to establish performance specifications for the assay itself. Okay, the components of the assay need to be qualified. There's a qualification phase, and you have to determine whether the components that you're going to use in your method are suitable for use in this particular assay. And a component of an assay is defined very broadly. A component can be an instrument, reagent, software, and even the staff that is performing the assay. All of those entities need to be qualified. And then you need to have quality control procedures to continuously ensure and monitor that the components of your assay and the assay itself is performing as expected. Okay, and FCS Express I'm going to show how FCS Express can help with different aspects of all of these types of components that are needed as part of your validation process. So before we start a new assay, we need to qualify the components that are used as part of the assay development. And as everybody on this call, I'm sure knows, this all needs to be documented to a great extent. One of the key things that we're going to be discussing throughout the course of this webinar is how to fulfill the documentation requirements that are onerous and extensive, and fulfill those in a streamlined manner that really doesn't take a lot of extra burden or extra time on you. So these procedures must be documented, must contain not only procedures that they're what the expected performance of the component is like, they must contain documented procedures to report deviations and what one does with the deviations. And these sort of procedures generally need to be run once upon the initial use of a component of your assay until the component changes, right? And it's up to the laboratory to determine what type of change requires a revalidation. So, you know, some examples of what would require a revalidation because there's a change, perhaps a new lot of reagent, perhaps new equipment, the hardware changed or the software on the equipment changed. And for software, perhaps a new version came out. Maybe you wouldn't necessarily change it just because a new version came out. You would look and see what features were modified in that version and change it based on the features that were changed if you use those features. So that really is dependent on, you know, the lab itself. No matter how you decide what to do, it is always time consuming and requires specialized expertise to write these validation documents and come up with the validation procedures. You need expertise in document requirement. You know, what do these quality documents need to look like and need to contain? You need expertise in the component being qualified. So, you know, you're not going to go qualify some complicated instrument if you've never used it before. How are you going to write those procedures? So you need to have somebody who really knows that component or that instrument or that device or that reagent to know what can go wrong, what needs to be tested. And you need somebody who has experience writing validation scripts to accurately write scripts that will comprehensively test the component that you're looking at. Okay, so this is all very, very challenging, very, very time consuming. And, you know, the average, you know, lab person might not have the expertise even how to do this, and you need specialists all working together. So in order to minimize and help with those requirements, FCS Express and de novo software do provide an installation qualification document. Installation qualification is necessary for hardware and for software, and it verifies the proper installation of a system. And we have a 50-page IQ that includes all the sections that are required for a proper IQ document. You can see a brief table of contents of them over here. They inquire scope, instructions how to execute the protocol. There are instructions here what to do upon deviations. There are references, there are attachments, and the document itself. Okay, all of our documents are, of course, approved and have places for you to approve them as you're going through the document. And here's an example of the document, or one of the documents. They all follow a similar role where every section has a purpose, what you are validating that is installed correctly, any prerequisites in this section over here in the middle, and at the bottom here are the actual test script that you follow. And every test script has an instruction, what you're supposed to do, an expected result, and here is where you put the actual result and whether that passed or failed the particular test that you're doing. Okay, so again, you don't have to be an FCS Express guru to know and read our website and read our documentation and figures out what makes a properly installed software and what not. You can just go through our IQ example. It takes an hour or two, and then you have a properly documented IQ procedure that you can run and rerun as you install it in new hardware or new instruments that will satisfy any inspection. Okay, and similarly, we also have an operational qualification document for you, which is an OQ, which is a, OQs again necessary for all hardware and software systems, an executed test protocol that documents that the system meets defined functional requirements. And this is generally used to test the general functionality of a software or a hardware. It doesn't, the OQ is not designed to test it in your exact specific scenario. It's designed to test that it is just in general working as expected in general. Okay, and so as you can see over here, again, we have a very extensive OQ that will survive any inspection at all, going through a lot of the key functionalities of the software, the layout functionality, plot functionality, gating functionality, statistical functionality, batch processing, data list, compensation, logging and security, etc. So every section, it just goes through the key functionality of FCS Express as it pertains to that section, and it will go through and test various aspects of that functionality. And again, they are, they're all in a similar format where you just have a script and you just follow through the script, and we can include layouts and we include sample data, and you just walk through and fulfill the script. One of the, I would be remiss to say that the IQ and OQ are a package that are for sale. They don't come for free, but they're very, very well priced if you consider the time and the effort that it normally takes to write your own validation documentation. These are just time savers. In addition, the IQ and OQ are both written such that you could perform them yourself in your laboratory if you wanted to. But we also offer a consulting service where we will send somebody out to perform the IQ, OQ on site in your facility as well. And that saves up your staff to be doing the things that they normally are doing. Okay, so again, one of the things that that de novo software provides to help facilitate your quality system. Okay, so that's to help qualify your software. Of course, you will have to come up with your own PQ. Since we don't know what assays you're doing, we can't really provide you with an out-of-the-box PQ. But at that point, we would presume that you're familiar enough with your own assays and your own things that you would be able to write the PQ, and you can even model them off of either existing PQ quality procedure documents you have already in the lab, or the IQ and the OQ documents that we've provided you. One of the other components, and arguably the most important component of any assay that needs to be qualified, is the staff who are performing the assay. Staff have to be appropriately trained. That training needs to be documented, of course. Once the staff is trained, the competency needs to be assessed periodically and documented. And while the qualification of the staff can be partially performed just based on their CV and experience, you do have to document and show that staff members were trained on the exact protocols and instruments and strategies that you are using in the laboratory. Okay, that training process is often very ad hoc, very difficult to document. It takes time and effort to come up with the training procedures, and it's just a challenge to continuously do. So how can FCS Express help? FCS Express can help by utilizing our SOP function to help do the training. So SOP, Standard Operating Procedures, is primarily actually designed to support what you guys would normally think of as SOPs, is to standardize an analysis in your lab, an analysis protocol. The SOP has sophisticated rules about what actions are allowed during a particular step of the SOP. So for instance, you can say in this step of the SOP, you're only allowed to move gate 1, but in that step of the SOP, you can only move a marker, and in that step, you can only remove a quad. So it's could be set up very granular about what is allowed during different steps of the SOP. You can also define your own criteria when users are allowed to go to the next step of the SOP. So for example, if there is a certain statistic that's out of range, you could say the user cannot go to the next step of the SOP until they document why they are allowing to proceed, even though this statistic is out of range or this value is out of range. Or you could just say, no, you can't proceed to the next step until you correct this error. It can't be out of range. So you have a lot of options in that way. The SOP instructions can include text and images and statistics and more. The document itself, the layout, tracks all the information about the actions that were done in the SOP, like who performed each step of the SOP, what time was it performed, what was exactly done in every step, and will automatically generate reports showing that each step of the SOP was completed and by who and when. So the SOP functionality is can be used for just a normal standard operating procedure, but it can also be used for training and competency. And this is what I'm going to show a little example of here. Okay, so we're going to launch FCS Express. And let me just bring it over to the right monitor. And so here is a training layout that is designed to teach people how to gate on lymphocytes, monocytes, and granulocytes. Okay, so this would be for like a novice technician who's just getting started in the lab. And it says, you know, right here you can see select quality in the ribbon above and run the SOP to get started. So we'll do that. We'll run the SOP. And you can see that what popped up once the SOP started is a little information here about lymphocytes and a little picture of the lymphocytes. And here are the instructions. Click on the red gate and move it to include the cells known as lymphocytes. You can change the gate shape to capture the whole population as shown here. And below at the bottom in that SOP instructions, it gives the user a picture of where the lymphocytes are. So we're going to follow the instructions and we're going to move the red gate to where the lymphocytes are. I'm not going to bother to change the shape, but we certainly can. And notice there's some red X's here to show that the lymphocytes are not yet gated correctly. And if I make this gate bigger, you can see that I've surpassed the criteria for when I've gated accurately on the lymphocytes gate, and we get a little green check mark. Now, I want to stress we did this as a very quick and simple layout. This is obviously not a perfectly gated lymphocyte population. You can choose which criteria and what criteria you want to use to determine if the population is accurately gated or not. Okay, so it's really up to you how intense and how accurate you want to be for determining if the user did the right thing or did not do the right thing. Okay, so now that I've accurately gated it, I'm going to go on to the next step. And now you see it change to say learn more about monocytes. Gives a little bit of information about monocytes and a picture of a monocyte. And it says position the green gate over the monocyte population. So I'm going to do that and move my green gate. And then I'm going to go to the next step. And now it gives a little bit of information about granulocytes and a cool picture of granulocytes. And I'll move my granulocyte gate like it's showing me in the little instructions over here. You can see that I've got 3 check marks. I've correctly gated my samples. And when I go to the next step, because I got 3 check marks, it gave me thumbs up and said that I passed my little training. I am now a gating expert. Okay, and there I completed my training. Okay, of course you can include this now and have the technician enter their name, enter their ID. You can have it put in the date, save this off as a PDF, and you've got a documented procedure, a documented record that, you know, a particular user completed their training for this. Of course, this is a very trivial example. You can extend this to training for your own specific assays with your own specific criteria. You can include data from normal cases and abnormal cases within your training layout, and the users would just go through and one at a time. You saw that things can appear and disappear, like those monocyte and lymphocyte and granulocyte text box explanations. Similarly, you can have plots appear and disappear, gates appear and disappear, and you can have the users do different things during different parts of their training protocol. Okay, now once they've done their training protocol, we can assess their competency at a later date periodically. And we can do that by opening up the competency layout. Okay, and here's the competency check that is performed periodically. And what you would do, again, a very simple one that we made to test if people are still competent in gating their lymphocytes, monocytes, and granulocytes. You would enter your name. And you notice here that this layout only has 1 page on it, the gating competency page. You can see that at the bottom of the document over here. Okay, there's only 1 page that the user sees, and this is because the user is logged in. I logged in previously as a technician. Okay, so the instructions for your competency test are enter your name above, place the red gate on the lymphocytes, the green gate on the monocytes, and the blue gate on the granulocytes, and then run the batch to save your results for review. So let me try to do that. I'm going to put my lymphocytes gate, and I'm going to remember that this is where they're supposed to be. I have my granulocytes gate, and you know, I don't have that great a memory, so I put it there. Sorry, my monocyte gate, and I'm going to put my granulocyte gate here. Okay, I finished my competency assessment. Let me run my batch to save what I'm doing. So I'm going to do batch, and then I'm going to run it. And then what happened is that when I ran my batch process, it saved my competency analysis to a file. And I'm going to show you that file over here. That file is on my desktop right now. And it's this layout that was saved. And so what I'm going to do now is I'm going to reopen this layout to assess the competency. And when I reopen the layout, instead of logging in as a tech, I'm going to log in as an admin. Okay. Now, again, in our security system, and if we activated the full security module, every user has a username and a password. They can't get into their account without knowing the password. For simplicity's sake here, I've just configured my system to not have the whole username password, to not have to continually enter passwords over and over. But rest assured that if you do purchase our 21 CFR with security, there is a proper security system as part of it. So when I open up the layout as an admin, you can see that there's actually a whole new page available. There is the gating competency page, which is what the user did, the technician did over here. But on the very result page, there is the a page that the admin can see that is review the results. And you can see here that the admin failed. It's telling the admin that they failed the monocyte test over there. Okay, and so the admin can choose to fail or pass the user depending on what they want. Here we're going to fail the user, this technician, and we are going to, you can see, run the batch to save the results with the PDF, and it will save these results as a PDF. And now you have the documented competency test for that particular user. And as you can see that you may have assumed, we actually just reused some of the same, the competency and the training layout are basically the same, just we hid a lot of the hints and everything like that in the competency version of the layout so that, of course, the person doesn't see them when they're doing the competency assessment. Okay? So again, you know, there's a lot of stuff in here. And you have a lot of flexibility to set up your criteria in any way you want, as stringently or loosely as possible. But it does give you a very nice streamlined way using your own data and using, you know, your own analysis and your own criteria to create very nice training and competency assessment layouts to support your quality program. And again, if anyone has any questions on that, please don't hesitate to type them into the question and answer section, and we'll be glad to help to answer those. Okay. So some other ways that FCS Express can help with your competency, with your quality program, is validation of qualification of your reagents. One of the things that is very important is antibody titration assay. You always need to titrate all of your antibodies before using them in an assay. The antibody titration, and also when you get a new lot in general. Antibody titration with other software is generally a very time-consuming process. You run your primary flow analysis in the one software. You either copy and paste or export or type in a bunch of the statistics in another software. You then might run a regression curve or something like that in the other software. You then have to copy and paste images to make some sort of report based on that, and document all three softwares together that you know how you arrived at your final titration values. In FCS Express, you can do all of that in a single software, and there's a beautiful layout here I'm going to show that was created by Rob Andrews at Lab PPM to show his antibody titration that he does, and it really can't be any easier to do this complex analysis. Okay, so here's Rob's panel. You can see it's a 6-tube assay over here, 6 different dilutions. You see all of the dilutions, each dilution is present over here. He gated on singlets and also on the population of interest. In fact, this is not just the raw statistics. If I scroll down a bit, you see that Rob actually calculated the stain index. So in FCS Express itself, which is the subject of an upcoming webinar that you can see in our upcoming webinars page, you can actually calculate the stain index right within the software based on the raw data. And then Rob is graphed the stain index against the antibody volume right here. And the layout itself now will automatically look and pick what is the optimal dilution to use based on criteria that was determined in Rob's lab, how they want to pick the optimum dilution to use based on the stain index. Okay, I do want to stress this is all live in real time. As I move my gates around, you can see that the stain indices are being recalculated based on the statistics of the gated population. The graph is updating in real time. And if you notice up here, the amount of antibody that is recommended is changing based on how the regression graph is going. Okay, based on the criteria that Rob has determined. Okay, you can see a lot of other stuff is being pulled out. This is a beautiful report. All these tokens are being pulled up at the top here from the list mode data file itself. And then at the bottom here, there is a way for you to type in that the staining pattern is acceptable, the titration is approved, and you now have both an analysis where you determined your appropriate amount of antibody to use, and in addition, you have a fully fledged document to support your quality process that you can now store either electronically or print it out and store physically in whatever documentation control system that you have that you use to support your quality process. And the beautiful thing is on a day-by-day basis, whenever you get a new lot, all you simply have to do is replace the files that are in this data list with a new set of files. All of the plots will update. Generally, you don't even have to adjust your gates, but if you do have to adjust your gates, you can certainly do that. And then you get a brand new titration, and you will have everything ready for that new validation. It might take you all of 45 seconds to validate a new lot of antibody, or at least do the analysis for the validation. Okay, any questions on that? Okay, great. If you do have any questions, once again, please put them in your question and answer section, and we'll try to get to them. Okay, another type of QC that needs to be done on a regular basis is when you get in a new lot of antibody, you need to do an antibody parallel test to show that the new lot of antibody is performing similarly to your previous lot of antibody. Again, that's often a very time-consuming and labor-intensive and manual process. But again, Rob put together a really beautiful example of how this can work in FCS Express, and I'll bring that up here. So here is an antibody validation that he did, and let me get everything set up on the right monitor. So you can see here, this is the reference on the left-hand side. You can see that there is nothing on the right-hand side yet because we haven't loaded our new antibody that we got. All I have to do is add in that new, oops, sorry, I added the wrong file. Okay, so here's the right file, the new lot. And you can see here that the new data file populated in all of the right plots. You can see that it pulled up automatically from header keywords in the header of the data file, the dates that the samples were analyzed, the lot numbers. You can see here at the bottom, all of these gray things, again, are lot numbers that were entered at the time of data acquisition that are put into the table here for reference. You can see over here, we've got statistics from the original lot and statistics from the new lot. You see here, there is the antibody acceptance criteria that the MFI of the gated target population of the new lot is within plus or minus 20% of the current lot. You can see the difference in the MFI is only 9%, and you can see that there is a pass. And all of these automatically determined, of course, like I showed you before, as you update your gates and your markers and everything, this will all update in real time. And all that's left to do is for someone to sign the approval either with our electronic signature system module, or print it out and sign it manually, or type your name in, whatever is you feel is appropriate for your sample. Okay, or for procedures within your organization. All right, so it couldn't be easier. You just simply load in the new sample, you see them side by side. Most of the information and the criteria is automatically calculated for and applied, and you're done your lot validation in literally 30 seconds or so, if all goes well. If you have a failing example, which I will show over here. So here I'll pull up a failing example. So here's our reference, and now again I'm going to add a new sample, and you can see what happened if we go to the bottom of this sample, that the MFI of the target population still has to be plus or minus 20. However, you see that the difference was 27%, and the layout automatically failed this sample. Okay, and you can double check the gating and make sure the gating is okay, and perhaps see if there's, just needs some gating adjustments, but definitely this new lot is a failure. There's really nothing unreasonable about the gating, and it's simply wrong. Okay, or it simply should be rejected. And you can then very easily get this report printed out, and you have a final report that supports the fact that this lot was rejected and you have to order a new one. Okay, so again, streamlining what is a very tedious, time-consuming, labor-intensive process. Okay, similar example for lysing buffer. Whenever you get a new set of lysing buffer, you need to validate that the new lysing buffer is performing similarly to the old lysing buffer. And a very similar type of analysis was performed. Here's the current lysing buffer at the top. I will go in and add the data from the new lysing buffer. Below. And you can see that it populated. There are 2 tubes. Even though there are 2 tubes, each tube knows what plots it's supposed to go to. And again, here are the acceptance criteria clearly documented within the validation document itself. Here are a whole bunch of metadata came up from either hand typed in or pulled up from the metadata in the FCS file itself. They chose to enter the review date and the review time manually. That could actually be automated as well to be this current date and this current time. There are certain criteria that can be automatically detected. So for instance, the viability and difference between lots and difference in debris must be between under 2%. So you can see here, those are automatically calculated over here. But then they also manually want to verify the light scatter pattern. They can manually choose if that pattern is acceptable or unacceptable. And you can see that even if I make one of them unacceptable, my final acceptance fails. Because in order for this to pass, Both the light scatter and the staining have to be acceptable, as well as the differences be under 2%. Once I make these back acceptable again, that they were visually determined to be acceptable, then I'm passing my validation. Okay, so again, not only are you doing your analysis in a very, very rapid manner, you're also immediately getting out a report that can be used to support your quality process. Okay, here's a more complex example of a cocktail pattern. I'm not going to go through this because it is not going to go through this in as much detail, but because it is a much more complicated. But you can see this is a multi-tube. There are all, 4 different tubes as part of this cocktail, and they all have to be validated against the previous tubes. And again, the most all I'm gonna, I'm not gonna go through it in detail, but all I'm doing again is loaded the second tube in right now. And you basically can see that the whole layout is set up to go through and compare. You see a whole set of pass-fail criteria for all of the different markers here are determined based on the rules in that lab. I do want to stress we never provide validation rules or canned validation rules for you. You need to determine what those rules are for your own laboratory. However, we can help you implement those rules and automatically check those rules once you decide what they should be. So here are the, again, acceptance criteria clearly listed as part of the validation document, and the determination of whether those criteria pass or fail are all automatically calculated by the layout itself. So again, I'm not going to go through this in detail. There's a lot of really cool things in here also that assess compensation, quality of compensation between the old and the new assay, old and new cocktail, and many, many things are also just automatically assessed. It is very quick to simply load a new set of data in, and then you automatically have a rigorous and validated document that you can again sign either electronically or physically and have as part of your quality process. Okay. One of the other unique things that we have are integrated live updating Levey Jennings graphs within your within the software itself. Many instruments do have Levey Jennings graphs that come with them that are usually used simply for instrument validation, and they're kind of hard-coded to your daily QC that you do on the instrument. With FCS Express, you can build Levey Jennings graphs of any statistic, anything that you want to track at all. And you can see here what we're doing is we're tracking the CV of different populations over time. And you can see that as I move my gates around, these Levey Jennings graphs are actually updating in real time to track the time course of the CV based on the gated population. And you can see in another set of graphs, I'm actually graphing the mean fluorescence of the different markers over time. And you can see again, as I move my gates around, it is updating the Levey Jennings plots. And here actually also showing in this little spreadsheet to the right, the raw numerical values that underlie those Levey Jennings graphs, with those values being color coded when they're out of control or in control. Similarly, everything about these Levey Jennings graphs are customizable. The out of control line colors, the number of standard deviations you want to consider in control and out of control. There's a huge number of customizations you can make to support your own particular quality process. So again, this can track any statistic on any marker, whether those be on cells or on beads or on controls. It can track scatter, it can track fluorescence. You just basically set it up to track whatever statistic you like. It can also track percentages within gates. So if you want to track over time, what is the percentage of the CD45 lymphocyte gate, you can certainly track that and ensure that that doesn't change significantly over time. Okay, there's also QC within the analysis. A lot of people here are using that already. That's one of the real benefits of FCS Express, and I think that that's widely utilized within sort of general usage in the clinical and validated lab. But just to give some examples very, very briefly, you can see here that on this report, the viability assessment that is located down here is currently good. Okay, but if I were to go and change my viability gate, which is actually on the next page, and bring the viability down and go back to that first page, you can see now that it's automatically updated to say that the viability is unacceptable. Okay, this is an incredibly trivial example of the QC criteria that you can build into your standard panels. You can build in alerts and criteria based on markers, based on ratios, based on percentages, based on, we even have people who have built alert in a multi-tube panel. You want to ensure that you've loaded the correct data into your panel, that all the tubes come from the same specimen. So there are alerts that check the keywords in all of the data files and ensure that the accession ID or the sample ID is the same in all the tubes that were loaded. There are so many alerts that have been made based on common errors that occur in the lab. I can't list them all here, but suffice it to say that any, pretty much anything that you can think of that you want the layout to automatically check for to support your quality program, to ensure that errors aren't made, we can build rules and give warnings and alerts when those criteria are met. Okay. Whoops, to go back to the presentation now. One of the other things that's a real strength of our software that's of course critical to more sophisticated compliance programs is 21 CFR Part 11 compliance. We have by far the most sophisticated 21 CFR Part 11 compliant functionality within our software that supports your 21 CFR Part 11 compliance program. If you are doing things now with your current software and printing out papers and signing them and dating them and keeping them in files and trying to cobble together some sort of audit trail or what could suitably, potentially, if somebody squints the wrong way, look like an audit trail, FCS Express provides an incredibly detailed audit trail that gives detailed changes over every single action that was done. Not only do you know that the gate was moved, you know exactly what coordinate it was moved from, and you know what coordinate it was moved to. Not only do you know the data file on the plot changed, you know what data file it changed from, what data file it changed to, at what time, by what user. Our permission system to support the 21 CFR Part 11 functionality has over 400 or 500 granular permissions where you can create whatever groups you want, and you can assign different people to those groups. It's a completely flexible electronic signature system where you can create as many different signatures as you like per document, and you can set rules which of these arbitrary security groups that you've made can set those security can sign those particular signatures, and under what criteria can they be signed. And very, very flexible and powerful 21 CFR Part 11. There are other software that purport to be 21 CFR Part 11 in the flow space, but if you even give a cursory look at what the actual functionality that is provided to you is, you will see that FCS Express is far and beyond anybody else in the business at the moment. Okay, so please feel free to reach out. We actually have webinars on the website at the moment that detail a lot of the aspects of our 21 CFR Part 11 compliance features. So in summary today, much of the analysis and documentation burden of daily QC can be minimized using FCS Express. FCS Express allows you to build the QC according to your own rules, whatever those rules may be. We can calculate any statistics you need, any statistics you want, ratios between any parameters you want, and create rules and alerts based on those. And by automating many of the routine tasks and many of the manual steps, you not only save time, as you saw, you get the titration results out in 30 seconds just by loading the data. You also get better quality because there's less spots for error, less spots for transcription error, less spots for user error, less anything that's less tedious and less annoying will generate a better quality system. All right, so I want to thank you all for attending today, and I hope that you've seen a lot of people here already. I see on the webinar are already FCS Express users. I hope you've been given some ideas and some inspiration to how to extend the use of FCS Express beyond your regular flow data analysis into supporting your quality program and making it easy for you to take the software you already have and make your life easier in many ways. And if some of this seems overwhelming and you're like, how I want to do this, how do I do this? Please just reach out to support@denovosoftware.com. That's SUPPORT @denovosoftware, DENOVO software.com. And we will be very, very happy to help you translate your vision of what you want your quality system to be into a layout that can help you realize those goals. Okay, so there will be a recording of the webinar, will be emailed to everybody who attended. So if you missed anything, you can certainly rewatch it at your own convenience. So if anybody has any questions, I'm done the formal part of the presentation here. So feel free to type in any questions that you have based on this, and I'll be glad to address them. One of the questions is, what does a Part 11 compliant report consist of? A Part 11 compliant report consists of whatever your validation team and group decides will fulfill the Part 11 compliant requirements that are put out by the FDA in the context of your own lab. So, we can't tell you what a compliant report contains. In general, a compliant report will have an extensive audit trail. That's generally a requirement. The analysis will be performed by people with experience and documented training to perform that analysis. So you have to do that. You have to ensure that the people are performing the analysis in the way that you are supposed to be performing the analysis, and different labs have different approaches to ensure that. And generally we'll have some sort of electronic signature confirming that this analysis was reviewed and is substantially correct. Again, different labs have different interpretations of when and what signatures are needed. The great thing is that whatever your requirements are and whatever rules you guys feel you need, you can implement those rules in the functionality that FCS Express has. Are there any other questions? Someone asked if they can get a copy of the titration layout. I can't give a copy of the layout that I showed here. Again, this was created by one of our customers for use in their lab, but we do have other sample titration layouts that are available on the web. I think we even have a webinar how to create them, and certainly we can help you get on a screen share with you and help you create one of your own. So if you just want to write us at support@denovosoftware.com, we can get you that information. And again, we're perfectly happy to help you implement them on your own. We are really very interested and support our customers in using FCS Express to its fullest potential. There's no reason why you should have a software that and only use 10% of it. So we're really, really happy to get you started. We won't build the entire layout for you, of course, but we will give you examples and work with you to show you how to do all the key things. And then it'll be up to you to just complete the layout according to your own requirements. Are there any further questions? All right, well, if not, I want to thank everybody for attending. Once again, you can email us at any time at support@denovosoftware.com if any questions come up afterwards. And I do want to thank you for your time and thank you for being an FCS Express customer, and hope everybody has a great rest of the day. Take care.